great to see everyone every Thursday morning to get a big seminar before we start the whole day today in Asian time in China. It's really my pleasure to introduce today's speaker, Dr. Matthew Ong, a rising young scientist and a leader I can look forward in microbiome science and computational biology. Dr. Om is assistant professor now in the Department of Integrated Physiology at the University of Colorado Boulder, where he leads the Integrated Microbiome Research Laboratory. Dr. Om earned his PhD from the University of California, Berkeley, where he laid the foundation for his multidisciplinary approach combining microbial genomics with system biology. He continued his research as a postdoc at Stanford University, where he refined his expertise in metagenomics and mucosal immunology, advancing our understanding of how microbial communities shape human health. His lab now's mission is to uncover how microbial ecosystems, especially those in the human body, interact with our immune, digestive, and nerve systems. A major focus of his current research is decoding the infant microbiome and his profound role in preventing autoimmune diseases such as asthma, allergies, and type 1 diabetes. Through omics technologies, including metagenomics, metabolomics, and innovative computational method development, Dr. Ohm's team is charting new territory in both microbial ecosystem and precision health interventions. In addition to his work on the human microbiome, his lab explores microbial dynamics in diverse environments, from soil and wastewater to hospitals, shedding light on how microbes continue to both health and environmental sustainability. Related work has been published in outstanding journals, including Cell Science, Nature Biotechnology, etc. That's really exciting. Please join me in welcome Dr. Matthew Ong as he shares his exciting research at the intersection of microbiology, immunology, and data science. Please, Matt.
Wonderful. Thank you so much, Paul. I'm so happy to be here and talking to you all today about some of the research we've been doing recently, specifically related to metagenomic immunoglobulin sequencing of IgA in microbes. As Paul said, I just started my lab at CU Boulder last fall, so it's kind of very exciting to be here and talking to you all. Just in terms of a very general outline of how my talk is going to go today, roughly there's two sections. The first section is a little bit more microbiology heavy, kind of getting into some of the work that we've done looking into the microbiome across industrialization gradients, with a particular focus on the infant microbiome. And then the second half of the talk will be much more immunology focused, getting into some of these techniques that we've developed to study IgA binding in the microbiome, and kind of some of the findings we found from that. And so, wonderful. I always like to start with kind of this image of the human gut microbiome, because I think it's very kind of illustrative of some of the processes going on. So what we're looking at here, this is a mouse gut, but if you were to look at a human gut, it would look the same. This green line here is a mucous membrane that kind of separates our microbiome on the top left from our own human cells on the bottom right. So these blue cells are colonocytes, colon cells, and the top left, they're microbes. And so I hope what you can appreciate from this is just how close in physical proximity these microbes are to our own human cells. There's this dense, diverse, complex, chaotic microbiome within our gut that's really remarkably close, just separated by this mucous membrane from our own human cells. And of course, this makes a lot of sense physiologically if you think about that. A major function of our gut microbiome is to kind of digest food. So there's a huge amount of nutrient flow essentially going from our gut microbiome across this mucus into our human cells. But it turns out there's a lot of kind of other information that passes from our microbiome into our human cells. I'm outlining just one process here. There's a number of different ways that our immune system senses our microbiome. But in one example, there are a number of different short chain fatty acids that are produced during fermentation by our gut microbes that are sensed by particular host cell receptors that we've uncovered the specific receptors in some cases that kind of lead to different responses depending on what the concentrations of these short chain fatty acids are. And so essentially, our body over evolutionary time has learned how to kind of sense these different metabolites produced by our gut microbiome to kind of integrate this data and to determine, okay, does this gut microbiome look to be in a good state, in which case, the host response could be something like, tamp down the immune system and kind of enter a regulatory state, or, does the short chain fatty acids make us think that the microbiome is out of whack, something is wrong, and so we should respond by inflammation and kind of, ramping up the immune system. Um, and so, this makes a lot of sense, but it kind of leads to this potential problem of, these receptors that sense our host, these receptors on our encoded in our genome that sense these microbes, again, they're hard coded in our genome. We can't really change them. Whereas there's this strong possibility, something I'm going to talk about, that our microbiome has shifted rapidly over the course of industrialization. So if you think about, for example, the hunter-gatherer lifestyle that we humans lived when we were, developing these sort of mechanisms to evolving these mechanisms to sense our microbiome, we were, a highly unsanitized environment, eating tons and tons of fiber, no processed food. It's a very different kind of lifestyle that likely led to a very different microbiome than the microbiomes that we all have today, impacted by processed foods, sanitation of the environment, antibiotics, et cetera. And so this idea that essentially the microbiome that our body has evolved to expect us to have a good microbiome could be a mismatch between what our microbiome actually looks like. And again, this mismatch between what our body thinks our microbiome should look like and what our microbiome actually looks like is kind of hypothesized to be at the heart of a lot of these chronic inflammatory conditions that are associated with industrialized countries. I mean, these are things like asthma, allergies, type 1 diabetes, food allergies. These are conditions that are far, far, far more prevalent in post-industrialized, industrializing countries as compared to more, non-industrialized countries that live lifestyles more similar to the lifestyle that humans lived when we kind of evolved these receptors. And so for this first part of this talk, I'm really going to kind of focus on kind of the impact that industrialization has actually had on the microbiome, with an eye towards kind of whether these changes could potentially explain some of these chronic inflammatory conditions that are really associated with the industrialized lifestyle. And so it kind of pains me to say this, but this is the only picture I'm going to show from this entire publication I did during my postdoc work. There's a large publication where a large amount of work, we really compared essentially the microbiome of industrialized populations. In this case, it was mainly a bunch of healthy adults from California, to non-industrialized populations. In this case, it was mainly this hunter-gatherer tribe in Tanzania, the Hadza hunter-gatherers of Tanzania. And we found, in short, a huge number of differences in the microbiomes between, these industrialized and non-industrialized populations. Just to kind of summarize them, you see far more diversity in the non-industrialized gut microbiome. That's in cases of bacteria, archaea, bacteriophages, eukaryotes. I mean, just all in all, the non-industrialized gut microbiome is far more diverse than we have in the industrialized world. You also see a lot of species that are only found in non-industrialized populations. So, for example, there's a number of microbes that you see pretty consistently across the non-industrialized populations across the globe, but that we really just don't have at all in the industrialized world, and kind of vice versa. There are some microbes that probably all of us have in the industrialized world that are essentially, you never see them in hunter-gatherer tribes. And so there's some presumably taxonomic shifts that have happened as we industrialized. And there's also other things in terms of we noticed that the non-industrialized gut microbiome seems to be more adaptive to oxygen levels and kind of things like this. But for the purposes of this talk, I'm when you think about the adult lifestyle and the adult microbiome, there's a huge number of lifestyle factors you can imagine impacting the microbiome and leading to these differences. I mean, I already talked about it, but fiber consumption, sanitation of the environment, things like this. But when you think about infancy, there's kind of a lot of reason to believe that infant microbiomes might be much more similar across the globe. Infants are born sterile or near sterile, and we essentially acquire our microbiomes from our environment after birth. I mean, some are presumably acquired like from our parents, but also from these other sources. And so, and the other thing is that infants consume, a lot of infants around the world, for the first 6 months of life are consuming nothing but breast milk. And so you have this scenario where, if even a baby born in California and in Tanzania, they both are born without a microbiome, and they're both having the exact same input of breast milk for the first 6 months of life. And so there's kind of this question of, at what point do these microbiomes diverge? And the other kind of related aspect to this series of questions is that in the industrialized world, there's been a lot of kind of research going into how microbiome accession happens. And so, for example, we know a lot about, and this kind of was depicted here, how when you're building a microbiome, kind of there's these first wave of microbes that come in and they kind of pave the way for this next wave of microbes, and you kind of have this community accession process where the microbiome kind of goes through these steps to go from a low complexity infant microbiome to a high complexity adult microbiome. But really, this has only ever been studied in the context of the industrialized microbiome. So, kind of a way of putting it is we know how to build an industrialized microbiome, but we really don't know what the process looks like for a non-industrialized microbiome. And so these are the types of questions that I'm going to talk about for this first half of the talk, is really kind of characterizing, to what extent the microbiome in infants diverge along these different lifestyle trajectories, and then potentially, does that have relevance for human health? And so to answer that first question right off the bat, you do see kind of rapid divergence of infant microbiomes. I show this figure all the time that I made, but it's actually one of my least favorite figures I've ever made because I think it's somewhat unintuitive, but I'm showing it anyways. But the main thing I want to point out here is that on the y-axis, what you're looking at is microbiome composition. This is not species diversity or anything like that. The only way to interpret this plot is the closer the lines are and the y-axis is how similar the microbiomes are. This is the first principal component of a principal component analysis for those of you who are in the know in the microbiome field. But so essentially what you see is that, over the first roughly 2-3 years of life, right after birth, infant microbiomes across different lifestyles are as similar as they're ever going to be. And then you kind of see this rapid divergence of industrialized microbiomes peering away from these non-industrialized and transitional counterparts. And so, despite the fact that you're still having, inputs of breast milk, you still do see this rapid kind of divergence. Something that is consistent across all these different lifestyles, though, is that within the first 6 months of life, you really see this domination by this group of bacteria called bifidobacteria. That's kind of what's shown here. If you look at this 0 to 6 month column, no matter what the lifestyle, you see a huge amount of bifidobacteria, this yellow bar. And this is especially interesting because bifidobacteria are a group of microbes that specialize in human breast milk. And so I'll take a little divergence to talk about breast milk for a second because I think it's unbelievably fascinating. Obviously a primary component of breast milk is for humans to provide nutrition to growing babies. And so if you look at like what are the components of breast milk, number one is water. Number two is like, carbohydrates like lactose. Number three is fats like lipids. But pretty soon after that, in a pretty large abundance, are these compounds called HMOs, or human milk oligosaccharides. And what these are is complex sugars. Some of them are depicted here that cannot be that don't provide calories to infants and instead serve as essentially food for the infant gut microbiome. And so, again, to say that another way, we as humans have evolved this mechanism to which we can essentially rob calories of growing infants and instead divert those calories to the infant microbiome, which to me is just such a strong evolutionary signal that the infant microbiome is important. If we're taking calories away from infants and feeding the microbiome, and the types of microbes that specialize in HMOs, in degrading HMOs, are these bifidobacteria. And so presumably, these are the type of bacteria that we humans like to see in babies to kind of set up a proper microbiome trajectory. And there's a lot of reasons why that could be. They tend to lower the pH of the feces of the gut to encourage other beneficial microbes to come in and prevent pathogens. But anyways, but bifidobacterium is somewhat of a broad group of bacteria. This is a genus level grouping. And if you start to get more specific about different species of bifidobacteria, you see pretty big significant differences in their ability to consume HMOs. What you kind of see here is we know all the different clusters that are needed to degrade HMOs. There's 6 different clusters here with a number of different genes in them. And the one kind of species of bifidobacteria that is the king of HMO degradation, the one that is can do it the most efficiently, and has all of these genes, is this guy called Bifidobacterium infantis in pink right here. And what you see is in these non-industrialized infants, so again, these are babies in hunter-gatherer lifestyles, you see Bifidobacterium infantis colonizing in huge numbers of these infants, whereas in the industrialized world, it's relatively rare. And the same thing is if you just look for these HMO degradation genes directly, you see these clusters are, super abundant in non-industrialized infants and are far more rare in industrialized infants. And then finally, even if you control for breastfeeding status here on the right, you see that even if you look at exclusively breast milk fed industrialized infants, far lower abundance of Bifidobacterium infantis as compared to, in this case, the Hadza hunter-gatherers that are also exclusively breastfed at this age. Yeah, so you see essentially this Bifidobacterium infantis strain that really specializes in HMOs. It's naturally acquired by non-industrialized populations, but it seems to be absent in the industrialized infants. And this is significant because this is actually one of the few, in my opinion, kind of probiotic strains that has a lot of kind of compelling evidence of being beneficial for humans. This was a study that was published in Cell a few years ago where they took 60 breastfed infants. They gave half of them a placebo powder and half of them EVC001. This is just a strain of Bifidobacterium infantis, and measured them 60 days later. What you see is compared to the control infants, the babies that got Bifidobacterium infantis had a significant reduction in a number of these different pro-inflammatory cytokines, kind of suggesting that having this strain around lets your body get out of this pro-inflammatory state and kind of relax, and seems to be something that, again, tamps down the immune system in early life. This is taking a tangent into the personal, but it turns out this is a probiotic you could just buy on Amazon. It has a different name now, but I had a daughter at the time. I was reading this paper and I was like, okay, I guess I'll maybe just like buy this strain and give it to my daughter. I live in a non-industrialized lifestyle. I did not assume that my baby was going to pick this up naturally. This is a picture of what my daughter looked like at 7 months old when I kind of had her microbiome sequenced. But the course of events here is at 1 month old, I gave my baby these probiotics. She was exclusively breastfed at this point. At 7 months old, I paid to have her microbiome sequenced through this commercial company, Tiny Health. So I just shipped off a little bit of my poop to this company. And the kind of remarkable thing is that if you look at her microbiome composition, it is so dominated by Bifidobacterium infantis. And I've done the analysis, and this is the exact strain that I gave her from this Avivo product. And so what this tells me is that even though most industrialized infants are not colonized by Bifidobacterium infantis, if it is introduced to a non-industrialized, or sorry, even though most industrialized infants are not do not pick this up naturally, if you give it to them, it kind of it could dominate. I mean, like, again, I had not given my daughter I haven't given her this probiotic for 5 months before I sequenced her microbiome, and you still just see it as totally dominated by this single strain. Again, 70% of her microbiome was just this one strain. And so this kind of suggests that again, if it's there, it'll colonize. But for whatever reason, in the industrialized world, we're not picking this up. And there's kind of a lot of reasons why that could be. Some people kind of hypothesize that because, especially in America and in Canada, we have this long history of essentially promoting formula over breast milk, that we kind of extincted our local population of Bifidobacterium infantis, and now we kind of can't pick it up. The other unique thing about Bifidobacterium infantis is that it needs breast milk to survive. You never find this bug in adults. So unlike a lot of other microbes you can kind of acquire from your parents, this microbe you seem to have to acquire from other infants. And so, to the extent that, you have a baby and you're not going to daycare, it's probably not interacting with other infants all that much. So maybe you're restricting its ability to pick up this strain. Whereas if you look at the Hadza, these babies are hanging out without diapers all together while the men go off to hunt. And so it's kind of easy to imagine you could get this horizontal transmission. But anyways, I thought this is a very kind of compelling evidence. And the other interesting thing about Bifidobacterium infantis is there's a paper that came out in Nature just last month, kind of showing that having Bifidobacteria in your microbiome when you get routine childhood vaccinations leads to significantly better kind of antibody titers and responses to these vaccines. And so this is just kind of a summary figure here. They found that this effect was true for a number of different bifidobacteriums, including Bifidobacterium infantis. But essentially, these stars kind of indicate that babies that had these microbes in their gut microbiome had significantly higher, significantly better responses to these routine childhood vaccinations. Again, suggesting that there's something about bifidobacteria, consuming this breast milk that really is kind of beneficial for the overall infant immune system. And if you start to think about, why would this be? Like, why would there be this one strain that really seems to be so beneficial to the human infant immune system? You can start to think about some of these somewhat out there ideas, maybe about coevolution. And so There's a number of documented cases of humans and animals of microbes and animals co-evolving together in these kind of long-term evolutionary relationships. Especially in insects, there's a lot of kind of insect microbe relationships that are very clearly coevolution. In the animal world, one of the most well-studied and characterized one is this Hawaiian bobtail squid. So this is again another example of coevolution where the squid has essentially evolved these specialized organs to recruit and feed and house this specific microbe, Vibrio bacterium fisheri, that colonizes um, these again special organs that the bobtail squid makes, and in return, these microbes light up at night when the squid is kind of going overhead, so it allows it to hide from predators, to not cause a shadow by the moon. But anyways, the point is that there is a precedent for essentially long-term coevolution between microbes and animals. This is not that much of an out there idea, and I kind of hypothesize that Bifidobacterium infantis um, has potentially co-evolved with humans as well. And so this is kind of a hypothesis that we're working through right now with some colleagues at Stanford, particularly Ben Good and Zuru. We've been working on not focusing on Bifidobacterium infantis, but looking at other microbes. Essentially, can we find genetic evidence that humans and microbes have kind of co-evolved over very long periods of time? So the way that we approach this is we essentially compared hunter-gatherer populations in Africa with horticultural populations, specifically the Chimani in the Bolivian rainforest over here, and essentially asked, we know that, for example, these two human populations that still live relatively non-industrialized lifestyles on kind of opposite ends of the globe, based on fossil evidence and kind of human genomic evidence, we know that these human populations split from each other somewhere between 15,000 and 100,000 years ago, and that's what's shown right here. And so we essentially said, okay, if we know how long ago these human populations split, can we also use all sorts of tricks to look at the microbial populations within the guts of these populations to determine how long ago did they split? And what you see is for a number of different microbial species, not only do the Hadza in Africa and the Chimani in South America share large numbers of species, but if you look into the details of the population genomics of these microbial species, you find that they have split times that really match remarkably well with kind of what you'd expect based on known human split times, kind of suggesting that, and the conclusion that this evidence really points to is that when 100,000 years ago, call it, when the Chimani and the Hadza were one human population, they and split, they have retained their gut microbes over this course of this 100,000 years, kind of consistently over time, kind of again suggesting long-term stable co-colonization for long periods of time, and the type of things that would be conducive or indicative of coevolution. And so I don't think it's that out there to, for example, conclude that, or to suggest, hypothesize, I guess is what I'll say, to weaken my language, that this kind of humans making HMOs to support Bifidobacterium infantis, and exchange Bifidobacterium infantis setting up specific physiological conditions in the gut that are beneficial for our immune system. I could see that coevolution being legitimate. And again, to point this out, we haven't looked at Bifidobacterium infantis yet. This is another study just showing it's plausible for other gut microbes, but I'm very interested in kind of exploring this idea with infantis as well. And so to kind of wrap up this first section of the talk here, I talked about how there's a number of inflammatory diseases that are associated with industrialization, and that there are very clear impacts of industrialization on the gut microbiome in adults. I've also showed you how infant microbiomes kind of rapidly diverge over the course of early life. I mean, they start off somewhat similar, but you see this rapid divergence within the first year of life or so. I didn't talk about this, but we also have shown that the initial microbes that start colonizing industrialized and non-industrialized infants are different, which is presumably why you set up these alternative developmental trajectories. And I did talk about how these different developmental trajectories essentially lead to different microbial compositions that seem to be beneficial. So, for example, the non-industrialized trajectory seems to be leading you to pick up large numbers of Bifidobacterium infantis, which are kind of shown to be beneficial for immune health, whereas this industrialized trajectory is leading you to kind of not pick up this Bifidobacterium infantis, which is potentially related to some of these chronic inflammatory conditions. And so, I just started my lab a year ago. This is most of this work that I did during my postdoc. I was trying to, look at this picture and say, okay, well, if my goal here as a lab is to essentially try and fight some of these chronic inflammatory conditions by modulating the microbiome, where can I have the most bang for my buck? And I would argue it's here in early life, where you can kind of have small microbiome changes that kind of set up these alternative trajectories that kind of lead to long-term stable change. And that's something I forgot to mention, but adult microbiomes are remarkably stable. I mean, by the time you or even like 3 to 5 years old, that microbiome composition is going to be staying with you potentially for life. I mean, you see long-term coexistence of these strains, long-term persistence of these configurations. And so it's kind of like getting to this stable state microbiome is a very critical thing. And I would also argue that human biology, um, again, if you look at where do we as humans put the most effort into regulating our microbiome, I would argue it's also in early life, specifically through breast milk. I talked a little bit about HMOs, but there's also other components of breast milk that I'm about to talk about that are regulating the microbiome. And so kind of this next question that I'm going to start approaching is, how exactly do we as humans, what processes do we have to promote beneficial microbes? And I think the more that we understand kind of how human biology is designed to promote beneficial microbes, we can kind of understand what's going wrong in the industrialized setting to potentially try and either boost these processes or kind of do some sort of corrective action. So again, I was going to talk about breast milk again. To talk a little bit more about breast milk, here is a kind of diagram showing all of the different immune-related components of breast milk. The ones that are most relevant to microbes are kind of circled in red here. I already talked about HMOs, these human milk oligosaccharides. These growth factors are really interesting. There's essentially signal ways for the tight junctions of our intestines to kind of relatively open and close during different periods of feeding, which is kind of potentially beneficial for kind of setting up our immune system. I'm not going to talk about that, but it's a very kind of interesting component of human breast milk to kind of regulate epithelial tight junctions. But one thing that I'm going to focus on a lot is IgA. So this is an antibody that is pretty prevalent in human breast milk. And that's kind of unique to humans. If you look at different antibody isotypes in other mammals, you see mostly they have IgG as their primary antibody isotype in colostrum. Colostrum is just the very early milk, like milk within the first days of life. It's called colostrum, but it's essentially still just breast milk. But colostrum is especially high in antibodies. And so you see that humans really do have a unique amount of IgA compared to these other mammals. And the other thing that I'll mention is that I was very lucky enough to kind of establish a collaboration with this mother's milk bank in Colorado. This is a huge operation of essentially collecting excess breast milk from mothers and kind of processing it and giving this out to hospitals. Kind of around the area. It turns out that having breast milk is kind of known to be super beneficial to especially preterm infants to prevent a lot of these kind of really acute microbiome problems, like infections and things like that. And so, yeah, for the next section of the talk, I'm really going to be focusing on, kind of the role of IgA in regulating the microbiome, that specific aspect of breast milk. And so to talk a little bit about IgA, I am primarily a microbiologist. I dabble in immunology and I'm trying to get more into immunology. But yes, IgA is, in my read of immunology, one of the most interesting and really not understood kind of antibody things going on. So for those of you who need a brief refresher in immunology like me, there are 5 different types of antibodies that we humans make. They're called isotypes. IgA is right here. There's IgG, IgD, IgM. But IgA is kind of unique in that it has this dimeric structure here. It's also unique in that it has kind of this ability. It has special components attached to it that essentially let it resist being degraded by our stomach. So it can essentially transit the stomach in breast milk and still be active in the intestines. And it's also unique in that it is secreted out of our mucous membranes. So most of these other antibodies are primarily, circulating in blood and stuff like that. But IgA, it turns out we're secreting vast amounts of IgA kind of all the time. Every day we produce more IgA than all of these other isotypes combined. And this is primarily secreted out of, nose, gut, essentially mucous membranes. But the other really interesting thing about IgA is that we understand kind of so little about what its function is. When I first started getting into immunology, I bought a textbook, like Immunology for Dummies, that was written in 2010. And if you read what it says about IgA, it's just like, yep, IgA, like all the other antibodies, it binds microbes, it kind of agglutinizes them up, it makes them all stick to each other, and then in the context of the gut, these kind of globs of microbes can be removed via peristalsis, and IgA is just binding pathogens and stuff like that. But there was this kind of seminal study, which I'm showing in the bottom right here, in 2018 in Science, that showed that some microbes actually benefit from IgA, and by producing IgA that targets this microbe is able to achieve higher colonization densities and better persistence in the gut. And turns out it's likely because it kind of binds the microbe and kind of holds it to the mucus, so it prevents the microbe from kind of being removed with the rest of the microbes through peristalsis in the gut. And so more recent kind of appreciation of IgA, kind of shown in the bottom left here, shows that there's a lot of different functions it can have in the gut. So definitely, elimination of pathogens is definitely something IgA can do. No doubt, if you have like a chronic viruses, clear pathogens, IgA does bind those and kind of, agglutinizes them up and helps remove them by peristalsis. But there are also these other roles where, for example, it's been shown that, sometimes IgA, rather than removing microbes, can just change their expression patterns. So for example, if you have IgA that is just targeting a flagella, you can just induce these microbes to turn off their flagella genes. So rather than eliminating the whole microbe, you're just changing the expression of these microbes to kind of prevent expression of these particular pathogenicity factors. And so IgA can function, some of which are bad for microbes, some of which are good for microbes. And then to make things even more confusing is there are a number of different kind of specificities that IgA molecules can have. So, when you're doing mouse experiments, if you remove T cells entirely, turn off the adaptive immune system, you still see IgA produced that can bind these proteobacteria and kind of other pathogen-looking things. So even without the need to kind of do antigen-specific IgA binding, we still produce this general IgA that binds pathogens in general. Other more specific IgA is definitely also produced. There are cells in the gut that kind of pull antigens and do the whole adaptive immune system thing and produce very specific IgA, sometimes to very particular to strains of microbes. But again, kind of showing over here these properties. Some all IgA clones seem to bind huge different numbers of species. Some of them are extremely specific to just like a single antigen. And so again, IgA can do a lot of different things. It can be produced by a lot of different mechanisms. And to me, this is just ripe for really understanding, what is going on, especially when you think about, how much IgA is in breast milk, and kind of, the evolutionary significance of having it there. And so kind of this next section of the talk is really going to be focusing on, trying to understand to what extent IgA modulates the human gut microbiome. And so, kind of the premier technique for studying IgA in humans, if you don't want to do mouse models and you want to just study, human populations, is, excuse me, IgA-seq. This is a technique that was developed by Noah Palm in 2014 in Richard Favell's lab. And essentially the way that it works is if you just get a regular fecal sample from a human, about 15% of the microbes are just going to be endogenously bound by IgA. So in most of our guts, again, 15% of our microbes are bound by IgA. So you can, have anti-IgA magnetic or anti-IgA fluorescent antibodies to kind of tag these microbes that are bound by IgA. You can put them in a cell sorting machine to sort out the microbes that are positively bound by IgA from those that are negatively bound. And then you can sequence these two populations with 16S rRNA gene sequencing. And so what this allows you to do is, just gives you a complete broad picture of the entire idea of what microbes in the gut are being bound by IgA at any given time. So this is a very fascinating and great technique. The drawbacks of this technique all really come down to this use here of 16S rRNA gene sequencing. This is a technique that essentially it just amplifies out and you sequence a single gene from all microbes. It's the 16S rRNA gene. And so in that way, you can get a rough picture of what microbial groups are present, but you don't have a lot of specificity into like the species identity, the strain identity, or things like that about these microbes. The other benefit of 16S sequencing is that because it essentially starts off with a PCR reaction, amplifying out this gene of interest, is it works with really low biomass samples like you kind of get from cell sorting. But again, the drawbacks here are that you're missing strain to species level resolution. You have no insight into the genes that the microbes have that are being bound by IgA. You can't do any kind of like SNP level analysis. There's some growth rate stuff I'm not getting into that you can't do. And so kind of the goal that we had approaching this question would be to, can we essentially, adapt this IgA-seq technique to, rather than using 16S sequencing, use metagenomic sequencing, where you're sequencing, the entire genome of all these microbes. So you can, you have a perfect species identity, you can look at the genes, et cetera. But the reason why that's more challenging than just running a different sequencing thing at the end is that metagenomics requires far more input biomass than 16S sequencing. And so, people have done back of the envelope calculations that it would take approximately 20 hours of cell sorting here for a single sample to get enough biomass to do metagenomic sequencing, which is just, totally unfeasible as soon as you get to a study of any kind of reasonable size. And so we kind of developed this technique in the Sonnenberg lab at Stanford. It was myself and immunologist Sean Spencer that really kind of spearheaded this effort, as well as Evelyn. Essentially, the way that we got around this biomass limitation is by doing magnetic cell sorting instead of flow cytometry. As I'll kind of show, the downside of magnets is you don't get as pure of a sorting, but the upside is you get way more biomass. And so, essentially the way that this protocol works is you get your fecal sample, again, endogenously. You don't have to add IgA, just within our guts, roughly 15% of the microbes are going to be bound by IgA. You sort those out with magnets. You sequence both the native kind of unsorted fraction as well as the IgA positive fraction. And then at the end, you do some kind of computational tricks to essentially compare the abundance of microbes in the IgA positive fraction versus the native fraction to get a very kind of accurate idea about the IgA coding of each microbe in the gut. So if you're all curious in this technique, it was recently published. So yeah, we have the protocol out and published and all of that. If you look at kind of the flow cytometry data, you can tell that this magnetic cell sorting is definitely working. You see, large enrichment in terms of the fraction of microbes bound by IgA in these, as assessed by flow cytometry, in the IgA positive fraction versus the native fraction. Again, with normal cell sorting on like a flow cytometer, the purity is like 100%. It's way better than we're getting by magnets. But this level of enrichment is definitely sufficient to essentially, by sequencing both the native fraction and the IgA positive fraction, you can, with this data, even though the purity isn't perfect, get a very good idea about IgA binding of all microbes in the gut, with the huge added benefit of being able to look at species level resolution. So for example, to pull out Bifidobacterium infantis and separate that from other bifidobacterias, but also to do things like looking at gene content or at SNPs or kind of this other stuff that I'm going to get into in this next couple slides. So kind of the first thing that we saw is that IgA binding levels are somewhat discrete. The way that you read this plot is that, okay, so we developed this technique, and the next thing we did is that we ran it on, I think it was 40 fecal samples from healthy humans in California that we essentially had lying around the lab. And so these are, again, healthy adult samples that we sequenced. And in this case, each dot is a microbial species in a specific human sample. And so there's thousands of dots here, but again, each dot is essentially a IgA binding level of a particular microbe in a particular sample. The y-axis is the IgA binding probability. This is a metric that we didn't develop, but is very nice for essentially measuring IgA binding strength. You can think of it as the percentage of microbes in the population that are bound by IgA. And so if you kind of sort them all, so again, this is just kind of sorting all of these dots based on their IgA binding probability, you see that there are a lot of, roughly 20% of microbial populations that are essentially completely unbound by IgA. Very few cells of those bacteria are bound. There's roughly 60% of microbes are lowly bound by IgA. So, some fraction of their population is bound by IgA, but it's certainly not all of them. And then there are some microbes that have very high IgA binding. So in this case, pretty much anytime you see a microbial cell from this population, it'll be bound by IgA. And then these kind of lines at the end here, these indicate microbes that are only found in the native sequencing. We don't see them in the IgA positive sequencing. These are microbes that we only ever see in the IgA positive sequencing. We don't see in the negative sequencing. So essentially like have infinity values. And so we kind of use this classification scheme to go throughout to explore some of these ideas. The first thing that you see is that these binding probabilities are significantly different across different bacterial species or bacterial phyla. For example, proteobacteria, this is a phylum of bacteria that tends to have a lot of pathogen-looking guys in it, and you see they're indeed very highly bound by IgA, as opposed to Bacteroides. This is a phylum of bacteria that tends to have a lot of microbes that are essentially hang out in the lumen of the gut and digest fibers and stuff like that. And these guys are very rarely highly bound by IgA and are instead often uncoated. The more kind of interesting finding that we saw here is that we wanted to kind of explore this idea of whether healthy microbes are being bound by IgA, health promoting microbes, which is a very, very difficult classification to make. I mean, to me, there's no way of saying, of classifying microbes as either being a probiotic or not without making somebody upset. And so we essentially, for the purposes of this experiment, we just considered any microbial species a probiotic if it's currently being kind of marketed as a next generation probiotic. And what you see is that these microbes, these microbial species that tend to be associated with health, which again we're calling probionts here, have significantly higher IgA binding than all the rest of these microbes. Again, kind of suggesting that in the healthy human gut, IgA is, it's not just binding pathogens. To be fair, there aren't really many pathogens in a healthy human gut, but it tends to be specifically binding microbes that are associated with health, which is interesting and potentially suggesting that this whole mechanism of IgA being used to promote beneficial bacteria might be more kind of widespread than is currently appreciated. This is a figure that I actually really love. I spent so much time staring at this figure to try and tease out what was going on with IgA binding over the course of this experiment. The way that you read this figure is each column is a different fecal sample from a healthy human adult, and then each row is a different bacterial species. They're kind of colored based on their different phyla, as shown up here, and then the each square kind of indicates the IgA binding of that species in that fecal sample. And so if it's white, that indicates that that species was not detected in that sample. If it's gray, it indicates the species was there and it had no binding. Yellow is low binding and red is high binding. And the way that I kind of look at this figure is if you just kind of lean back and squint a little bit, you kind of see broad consistency in the patterns across these different samples. And so, for example, over here, this is a group of microbes that pretty much whenever you see them, they tend to be highly bound. Over here is a group of microbes that pretty much whenever you see them tend to be lowly bound. And then over here would be a group of microbes that pretty much whenever you see them tend to be unbound. And so what this suggests is that, you know, each human does not have a completely unique and specific IgA repertoire. There seems to be broad kind of consistency in the IgA responses that healthy Californians are mounting against these different microbes, kind of again suggesting consistency in it. And the other thing that you see if you look at this figure for a while, like I have, is that the microbes that are highly bound tend to be microbes that are associated with with health benefits. I mean, just being in the microbiome field for a while, you start to get a second sense for, is this a bug that you want in your microbiome or one that you don't want in your microbiome? And the ones that tend to be highly bound are ones that you would want in your microbiome. These are like healthy looking guys. And so again, kind of like that last figure I showed with probionts, it does seem like IgA binding in healthy humans is preferentially towards certain groups of microbes and especially towards microbes that are being marketed as probiotics and potentially health associated. A couple of other kind of vignettes from this story that I thought were particularly interesting. One is we had this question of, because we have the gene content of all of these microbes that are bound by IgA, we had this question of, can the genes predict IgA binding? We trained a machine learning classifier to do this that worked pretty well. Essentially, we had it's not perfect, but pretty good classification accuracy at predicting which microbes have high binding or uncoded binding based purely on their gene content. So essentially the input to this classifier is just a long list of genes and whether or not that microbe has that specific gene. But kind of the more interesting thing about this machine learning classifier, in my opinion, was shown in C here, which is essentially we trained classifiers to predict microbial binding based on microbial taxonomy. That's the green bar. And so that's essentially, okay, this is E. coli. That would be taxonomy, or on the gene content, kind of like these ones and zeros that I talked about for every gene, or on both the taxonomy and the genes. What you see is that the predicting on the genes does better than the taxonomy, but predicting on both does no better than genes. And so what this tells me is that the fact that you can see these broad taxonomic patterns in IgA binding is just because taxonomy and genes are correlated with each other. It's actually the gene content of these microbes that is determining their IgA binding content. And that taxonomy is essentially just a way of getting at the gene content. And so of course, once you have this, there's this question of what genes are most associated with IgA binding. That's kind of shown in this plot D here. The way that you read this plot is each dot is a different microbial gene. The more far right it is, the more associated it is with being bound by IgA. the more far left it is, the more anti-associated it is with being bound by IgA, and then the higher it is, the more statistically significant that association is. And so if you kind of do this plot, there's one gene here that tends to shine above the rest as being especially significantly associated with IgA binding, and this is the gene GH101. So this is a glycosyl hydrolase that is uh, that is used to degrade human mucus. And so what you see is that a large portion of the microbes that encode GH101 are bound by IgA. If you look at what microbes have GH101 versus those that don't, you see that the microbes that have it significantly higher IgA binding. And also, if you just kind of compare different glycosyl hydrolases. So these are genes that are used for breaking down carbohydrates. Microbes that have glycosyl hydrolases that are associated with breaking down human mucin genes. So again, our mucus are significantly associated with IgA binding. And so, the way that I interpret this data is not that this GH101 is like an antigen that IgA is binding. I think what's happening is that this is kind of telling us that the microbes that have a lifestyle that is involved in getting way up in that mucus barrier that I showed early on in that green zone, are the ones that are inducing a particular IgA response. And so, the microbes in the middle of the gut, these Bacteroides that are just kind of degrading the fiber, they're more or less not being targeted by IgA, but these microbes that again are degrading our mucus and right up against the mucus barrier, kind of closest to our human cells, these are the ones that really tend to be bound by IgA, which is a pretty interesting conclusion from all of this work. And then finally, in this case, I said that we ran IgA-seq, or this MIG-seq metagenomic IgA sequencing protocol on some fecal samples we had lying around in the lab, but these samples were actually part of an experiment that the lab did that was generated an immense amount of immune profiling on all of these subjects. And so, for example, for each of the subjects that we had fecal samples from that we ran this technique on, and we had blood information on them, so like triglyceride levels, et cetera, cytoph data on all these different immune cell populations. We had detailed dietary information data, O-link data on different chemokines. We had proteomics of the stool. We had short chain fatty acids of the stool, metabolomics of the serum, metabolomics in the stool. And so the naive question we asked is, okay, we have all these different measures of kind of human immune status. Are these more associated with essentially IgA binding of microbes or on traditional relative abundance of microbes? So what you see is that there's way more statistically significant association between the IgA binding of these microbes and these different measures of human immune status than there are between the regular relative abundance measures that you typically see. And so what's significant about this is that, if you're running a microbiome study and you want to know, are there microbes that are associated with some particular, immune status marker? The typical way of studying the microbiome is by sequencing the feces and then looking at the abundance of all the microbes there. So that would be this relative abundance measure. But if you do IgA sequencing instead, you see far more associations between these microbes and the host immune status. So essentially what this tells us is that the IgA coding of a microbe is much more indicative of kind of its association with other immune system things going on in the human than as opposed to the relative abundance of that same microbe. Kind of again pointing to the benefit of measuring kind of this IgA probability, IgA coding of these microbes. So finally, kind of wrapping up this talk here, all of this work was done in humans. Or sorry, it's also in humans, in adults. As you can kind of tell, hopefully I have a very strong interest in infants. And so kind of something that I've been working on, most of this work was done during my postdoc at Stanford. What I'm really working on now in my own lab at Boulder is kind of applying some of these techniques to infant gut microbiomes to kind of understand how the IgA in infancy kind of functions. And so in particular, we're kind of launching this study with a professor, Minghua Tang at Colorado State University in Fort Collins, kind of focusing on the running this MIG-seq technique on infant samples at 5 months old and 1 year old that have been randomized into a number of these different diets. And so again, these infants are recruited at 5 months old into the study. We have a baseline fecal sample there. They're then randomized into one of these different dietary groups to essentially have these diets complementary to breast milk. So infants are still consuming breast milk along with these other dietary groups. And we're essentially trying to determine among other things, whether these different diets have an impact on kind of the IgA coding and other microbes in the gut. And so the kind of first hurdle we face with this is, will this even work on infant gut samples? Again, we've only ever tried this in adults before. This plot here kind of shows IgA production in infants over the course of life. And what you see is that when infants are first born, they produce very, very little of their own IgA, and there's kind of a very slow ramp up before they produce as much IgA as adults. So this is kind of infant production of IgA. You see it is very low at 5 months of life, and is a little bit more at 12 months of life. This other subplot up here is showing IgA concentration in breast milk, and that shows the opposite trend, where very early in life, colostrum, this has very high levels of IgA in breast milk, whereas kind of as you get older in life, there's less IgA present in breast milk, but there's still some present. And so we kind of had this question of, okay, well, is there even going to be enough IgA in the infant gut for us to detect if microbes are bound by it? And the answer is yes, thankfully. This is work that is really spearheaded by my graduate student, Jing Huan, who started with me last fall and has been fantastic. What you see is kind of, this flow cytometry data, as you expected. If you don't add the anti-IgA antibody, you don't see any IgA binding. You add native IgA, you see some. And then when you enrich for it with magnets, you see even more. And kind of also encouraging is we see that there's a strong difference by time point. So very early in life, when infants aren't producing their IgA, you see very low levels of IgA binding, whereas by 12 months of life, where infants are producing significantly more IgA, you see a lot more IgA binding. So that's encouraging. We're still working on experiments to kind of parse out what fraction of this IgA binding is from breast milk versus endogenously produced by the infant. And then finally, the last kind of data slide I have to show is that we are seeing a significant difference by diet as well. And so in both infants that are randomized into this meat group, in this plant group, you see at the baseline measure before they're on the diet, similar levels of IgA binding, whereas the meat group, you're seeing significantly higher levels of IgA binding by 12 months old as compared to the plant group. And so, this is still preliminary adults, and we're still preliminary results, and we're kind of chasing this, but it's kind of exciting stuff to see. And so in conclusion for this section, I hope we've convinced you that this technique we developed, MIG-seq, can reveal kind of species level IgA binding patterns as opposed to previous techniques that are really kind of focused on more 16S broad taxonomic groups. Kind of the main finding from this work, in my opinion, is that the microbes that tend to be bound by IgA are really these microbes that are, associated with the mucus, really kind of very close to human colonocytes. They also tend to be these probiotic species that are, associated with, generally associated with immune health. Again, that's a hard claim to back up because the microbiome field is so squishy about, is this a good bacteria or bad bacteria? But in general, they seem to be good bacteria. And then finally, part of the reason why I'm so excited about IgA as a research direction is because there's a lot of ways that you can imagine applying it in a therapeutic setting. So for example, when you give vaccines, some of the first antibody responses are IgA. So you could imagine kind of designing specific IgA vaccines, IgA monoclonal antibodies. You could imagine, doing some of this stuff with both the mom to kind of induce specific IgA in the breast milk. You could imagine doing some of this in infants to essentially try and ideally promote colonization of beneficial microbes by essentially, designing IgA through some of these mechanisms to essentially encourage engraftment. And so I think that there's kind of an exciting potential for next steps in this avenue of research. And so with that, I kind of acknowledge my lab. This is us here. Here's Jing, me. There's my daughter Kira, kind of more grown up. My beautiful, yeah. And then I'd also like to acknowledge my former PI, Justin Sonnenberg, as well as Matt Carter, who was a graduate student in the lab with me that did a lot of the work I did there. And this is my beautiful lab in Colorado now. With that, I'd be happy to take any questions.
Wow.